Bottom in sight,
18.
I think.
I think it might be. That's lens flare.
Yeah.
Yeah, co-pilot, I don't.
Pilot has bottom in sight,
Bottom in sight.
8 meters altitude,
Is
coming
that
down
enough?
slow.
Oh, I was watching the sweep of your sonar.
Could you, when you get a chance?
Yeah. I don't see When it. when it first came up, it looked real, but now that it's swung all the way around.
You're landing on a soft sedimented bottom.
Yeah, I think I'd like to hit that white balance again.
Oh, yeah? Okay.
Yeah,
Do you want me to
sorry.
get that in position?
Yeah, get that in position. I'm going to go ahead and get over to the, what's our line of advance? It's going to be now.
255.
255. Yeah, I got to come around to
Copy.
side
Oh,
here.
this looks silty.
Style.
Bridge, ROV nav.
Hey, we're just getting settled on the bottom. Our depth here
ROV
is
nav, bridge.
2899
Bridge, the
meters.
ROV is on the bottom.
Roger that. ROV on the bottom.
Yeah, video, we have bottom in Sirius too.
Yeah, the sediment here does look fairly compacted.
Did you take some lead off, Chris?
No.
Yep.
Well, when you get a chance, Mike, could you read off temperature, salinity, and DO for me?
Indeed. So temperature is 4.08 C. Salinity is 34.97. And
like the
the
lights are in a
oxygen
strange
is
configuration.
They are at the moment.
7.50 milligrams per liter.
Thank you.
Copy that. Coming down. At 18, I'll come down to 15.
I'm coming towards you now. I think we'll be all right.
Copy. Yeah, let me fix the video, the lights for you video.
Thank you, Amanda, for adding that to the event log. Okay.
Just pass over a really blue
Ooh, that was
sea
an
star.
interesting sea star. We,
I'm
Side.
sure you're
we're kind of flying
Give it
by,
color.
catching up.
Yeah, I'm trying to get into formation
Okay.
here.
Looks like there's a lot of plant detritus on the seafloor as well. When we get settled, we'll have a better look at the sediments before we get a move on.
Thanks, Amanda, for all those comments. We'll chat a little bit more about geology when we get to the rocks.
Adjust the sonars a little bit, pilots. Copy that. Thank you.
Pop into auto head here before things get spun around, Chris.
change some settings while you're doing it.
No two pieces of seafloor have looked the
line up
same
and then-
throughout this whole expedition. Even the soft sediment.
Indeed.
This looks more compacted than other places we have observed. There's a lot of detritus. Maybe some shell hash on the sediment surface as well. And then it looks like a large shrimp.
All right.
I think some
Tilting.
confusion on the genus here
Full zoom.
of that shrimp. We're doing another white balance.
Zoom
Might
in a
have to
little
come
more.
down a little.
Yeah.
Just to get it more square.
Yeah, just
Right.
bump it down a smidge. Oh, yes, that's it. There.
Come out just a little bit.
Trying to come left for you. Boom.
Maybe work with that.
Yeah. You might have
since the arm's out there.
And we
you might have
just
to adjust
reached
your
the bottom
Z-bias.
of
Yeah,
Exocet
little bit.
Seamount. We're at the base.
A lot of
We're going
shimmering.
to move towards the Seamount here in a little bit.
How's the
just
light?
white balancing, caught up with
Flat.
Sirius. Gonna, before we move, take a good look
Flat
at the
balancing,
sediment,
tapping.
do a zoom, and then move towards the slope.
I have auto head in at 240. Sorry about the spooge. Let me.
Speaking of Exocet
Co-pilot
and
keeps
flying
coming under
fish,
255.
I asked.
255. Ah.
I think I was talking to Art Howard the other day if he has seen any interesting life at the surface of the ocean, and I've seen a lot of sargassum floating around, but he told me he observed lots of flying fish, not lionfish, flying fish, but I haven't seen any.
I found two.
Nor have I. I've enjoyed the brown boobies though, diving off the bow and fishing.
Yeah. You found two,
White.
Dave?
Two dried up flying fish on the deck. It's been a few days though.
Oh
Yeah.
yeah,
What are the? What's on the bow? I haven't been up there.
brown
in 3
boobies,
weeks.
Yeah.
Brown boobies, cool.
and they sort of hang out on the lee side of the ship, just float, and they wait for the ship to kick up flying fish,
Ah,
and then they chase the flying fish. It's pretty great.
what am I doing back there?
Can't see them from the workshop.
Is that
Video's happy with that. Thank you.
okay? Is that a better one? You think?
Yes.
Okay, great. I will stow.
We're all set on the white balance?
I think so.
All set on the white balance.
So before we move, can we just get a zoom in of the sediment?
Certainly.
Thanks. Maybe all of the different detritus and things on the sediment.
Wow.
Yeah, hardly kicked up any sediment as we settled down.
We can zoom.
Assuming.
It's like some trash too.
Indeed.
What is that?
Looks like a tomato can
Yes, steel.
Yeah.
or applesauce.
Steel
What does it say?
can, Corroded. S in.
Simons. So we're looking at a piece of trash here. Yeah, it looks like a tomato can. Trying to read it. And there's some sargassum around it. A lot of pteropod shells at the surface.
So
Shilling secure. Sorry, now.
I was just going to remind watch leads that we have developed some expertise on the sea poke,
Okay.
if there's geological interest in that.
Okay.
So
Why
can we maybe tilt down just for
Let's try.
sediment when
You want
watch
to sea
lead's
poke?
ready?
No?
Yeah, why not?
Can we sea poke now while we're sitting here?
Sure.
Thanks. So in view right now are a lot of pteropod shells, and these, of course, they're swimming mollusks. So these animals are swimming in the mesopelagic zone in the pelagic area, and then eventually they fall down to the sediment surface, and what we're seeing is the shells or the tests of the pteropods.
Okay, come out if you want to do your sea poke. Not a flare.
Yeah, that's a swing arm. Sorry. That's the uppers. Decks on. do a little warm-up calisthenics here.
So I'm noticing
like you got the
really big reflection in the
Co-pilot, you got the
video.
port lower all the way around by the shelling.
That's not right.
Okay. Yeah.
Might need more pressure, Dave.
That's a poke.
Oh, that's what it was. Yeah.
You want me to bump up pressure? Copy that.
So what just
Rookie
happened?
mistake. We engaged the arm with insufficient hydraulic pressure.
Okay.
We had it turned down for energy conservation reasons. And like a pilot without coffee in the morning, it just couldn't rise to the occasion.
Yeah, thank you, Ned. Correct.
There's a brittle star off to the left.
Well, that gave us some idea how thick the sediment was.
Yeah.
You could just rotate. Or come out a little bit if you wanted to. I think
It's also a good way to test the current.
Indeed. All planned.
There's lots of advantages to this technique.
Yeah, there's not a lot of current
So,
here. What was the
co-pilot.
surface current
Yes,
now?
sir.
Need,
There was
left
not much surface
monitor-
current either.
Left monitor, starboard, manip cam.
On the launch, it looked like there was maybe a little bit of current from the northeast, but not much.
Anything in right?
That's good.
I can bring a mini Zeus around if you'd like.
Yeah, that'll be helpful when we get to poking around here.
You can see currents roughly west to east coming back at you.
Right now at the surface or at the bottom, west to east.
Yeah, we're facing
Yep.
west, and it's you can see it's kind of coming back at the vehicle. So roughly,
Looks like we made a little snow angel.
get a better one.
Snow angel.
Let me know if you need a different view.
Okay, left monitor, Zeus plus, please.
And the sound effects again today, team?
I was thinking a Harry Potter spell.
I don't know.
I
if we
think
can
that was
live
Todd
up
and
to that.
Jeff.
Yeah,
That was Todd.
Todd was impressive there.
So we're just using this sea poke, we're calling it, to measure the depth of the sediment. That's about the poke is 1 meter in length, correct?
80 centimeters.
Oh, 80 centimeters. Okay.
And
That's
it's
strange.
it has 5 centimeter gradations on it, so
Okay.
if we can
I'm
count
gonna
them if it goes in.
Spin a look at you, Dave. Lose our heading.
Yeah, it's okay. Probably need a little bit more zoom on Zeus
So we're
plus.
just testing how thick
Yeah,
the
good,
sediment
yeah.
might be here. We've done this in a couple of places. On top of the
Coming in.
pinnacle yesterday, it was only a couple
Might
of
need to
centimeters
expose
thick.
a
It would
little
barely
bit.
go
Yeah.
in.
The marks on
The
it.
day before, we could lose the stick in the sediment. It was so thick.
There you go. Does that help? Any light?
And
Okay,
You can
so
see the gradations, and they're 5 centimeters apart.
5,
That's
7,
10. All right. 15,
20.
20, 25, 30.
Oh, a lot thicker.
It's going to go all the way. There's 70.
Fairly compacted, but fairly thick here.
Yeah, so at least you probably could go further, Dave. Probably, you think?
Zoom on Sirius.
well,
if
if
you like.
I push the arm down any further, I'll lift the vehicle off the bottom. So,
So
yeah, no resistance felt
That's good.
whatsoever.
right. So at least 75 centimeters thick. Thank you, Dave.
It's a cool shot from Sirius.
We're just giving you practice with those manipulator arms.
Okay,
Yeah,
zoom
thanks,
out
Ed.
on Zeus plus, please.
All right,
Zeus
so
plus
I
zoom
think
out,
we've
aye.
got a good measure of the thickness of the sediment. have seen a lot of sargassum detritus in this area surrounding the seamount. A little bit of trash here, a tin tomato can in view, and a lot of pteropod shells. And it's interesting that pteropods are now considered to be in two separate taxonomic groups. So pteropods include sea butterflies, swimming pelagic sea snails, and sea slugs.
Thank you, team.
In the quiver, and I need, I think, mini Zeus on. Oh, wait a minute, there it is. Wow, good.
Read your mind.
Good, mate.
Just putting the sea poke back. We're at a depth of 2,897 meters in an area surrounding Exocet Seamount, and we're in a minute here going to take off and go explore Exocet Seamount.
So Dave, just to explain, there's a quiver that we can't see and then a magnet maybe on the top of the...
Yeah, they can't see it on shore. They don't have these views, but we have the left screen is a camera that looks from the right side to the left.
Mm-hmm.
The right screen is a camera looking straight down, and that's a rubber ball with a magnet. And out of view on the left, there's a tube that the tip of the poke is guided into, and then that holds it and slide it in with the magnet in place.
Thank you, Dave.
Stowing it inboard.
Stowing inboard.
Yeah.
There's a good number of things that are tumbling along the seafloor from starboard to port. If you look out, this little
It's off.
Yeah.
Finished
black
film.
objects.
Okay. Just poking along here.
Oh, you got a nice
And there's a
whirling
brittle
dervish
still there.
on port side.
Oh yeah. Where away?
255.
255I.
They come back.
Okay, we're on the move.
255. And fix your lights. And your other lights.
So we're at a depth close to half of where we were a few days ago in the Puerto Rico Trench, to the deep dive to 6,000 meters with the D2. So that means we're also at about half of the amount of pressure that we observed, well, not observed, but that we were at during the Puerto Rico Trench dive. And high pressure and cold temperatures often found in deep waters really do control the distributions, the bathymetric distributions, or the distribution over certain depth ranges of certain organisms. But organisms do have several adaptations to counteract the effects of high pressure and cold temperature. Both high pressure and cold temperature can decrease enzymatic activity, can lead to proteins unfolding and denaturing, can reduce the fluidity of membranes, the cell membranes. So animals have built some adaptations to counteract these effects, including having more unsaturated fatty acids in the cell membranes, including having more what we call protein chaperones to help with stabilizing proteins. Some organisms can increase the concentration of enzymes and things like that.
You're right.
Got it.
So the animals that we see living in deep waters have evolved several ways to be able to survive and flourish in low pressure, or excuse me, high pressure, low temperature environments.
It's very bright.
Zoom. Trying to get off the bottom. Get out.
Another
Just looking.
piece of trash, or is that a shell?
Coming out.
Oh, sorry.
Good morning, Santiago. I'm glad you're joining us from Toronto, Canada.
You're way out there, Dave.
Yeah.
I can come out.
Okay, I'm going to have to stay way off the bottom.
I think
here.
it was
Yeah.
trash.
Go gently because we are in
It's Siltville.
Spooge City.
Dave, I like some of your descriptions.
Everything I know about the seafloor I learned from the back row.
Okay, both your lowers are slightly deployed.
Got that?
Mini-Zoo's back home.
So thinking again
Ready
about
to-
adaptations
Oh,
to
yeah.
yeah.
Way ready.
Way ready.
Adaptations to high pressure and low temperature. So a lot of
Bridge,
adaptations we
Arvina.
can't see directly, right? These are molecular adaptations, cellular adaptations.
ROV
There
nav,
are
this is
also
Bridge.
morphological
Bridge requesting a
adaptations.
ship
So
move.
there's some fishes that have
100
in shallower
meters,
waters have swim bladders,
255
but
degrees,
in the deep
0.2
sea,
knots.
they have lost their swim bladders. Some other fishes in deeper waters have substituted air and swim bladders for lipids. And then
Roger,
there are
100
some
meters,
fishes that have more water
255,
content in tissues.
0.2 knots.
So,
Good copy, Bridge.
and then of course there's a suite of other adaptations for low light.
Boy, your birds are starting.
Hopefully
up a
we'll
ton
see
of
some of
stuff
those
here.
animals. We can talk about that more
It's
during this dive.
it's my wake, Ed. Not so much the birds.
Really?
It's just, yeah, the
So I
pressure
think we
wave
still
of
have Tim Shank on the phone
pushing
with
the
us. Has
vehicle
anybody
through the
else
water.
called in? Is Mike Ford still on the phone with us?
Not even sure we're actually starting up the sediment as
Yeah,
much
Tim
the
is
stuff
still
that's
here.
on top of it.
Yeah, agreed.
Glad you're with us again today, Tim.
It's very nice to be here, as always.
And you were able to see most of the dive yesterday, although
Yeah, it was a fantastic dive. Unfortunately, I was in meet and could
That's
only
it.
watch it and listen a little bit. But I thought that the diversity was just spectacular. And I always look at the inverts associated with the corals and sponges, and I thought the diversity was really nice. And in part, that was based on the diversity of corals that we saw. I think you mentioned 26 species, I think, yesterday, which was just, phenomenal. And the variety of associates we find on there, some very specific to those
Tug.
certain corals.
Sorry.
Thinking about
Sorry.
the crabs and the shrimp that we saw. Even saw the hermit crabs, which are
yeah.
much more rare that we see on these certain species. So I was just in delight. I've been going through the fame grabs overnight and looking at all the things. It's just spectacular. It's taking a little while to digest that, but that was really a one for the books yesterday.
Yeah, I was really impressed with the diversity of the squat lobsters, too. Several new morphotypes that we haven't observed yet during this expedition. I don't know if you had a chance yet to see the one that was camouflaged with the madrepora, so it was light pink. It really matched a madrepora stony coral colony. It was so cool. I've never seen that one, at least.
Tug.
Yeah, I know.
I thought that was absolutely spectacular. I did go through the literature trying to find it, 'cause I have not seen that one before.
Okay.
That morph, and I think I found a paper from 1964 discussed that morph in nearby, not this particular Puerto Rican region, but in the Caribbean at depth, 1,000 meters.
Very cool.
Anyway, very interesting.
Nice.
Yeah, we'll see what we find today, too. We're at a much deeper depth today at 2889 meters. Yesterday we were in water depths of 870 to 820 on one mound, and then we had came up in the water column and moved 1.2 kilometers to the southeast and surveyed another mound at about 925 meters, I believe.
And just chiming in very quickly, last night we carefully re-looked at the bathymetry that we remapped, and processed it, and we saw one other interesting thing. The first mound was not circular. It actually is very angular. It's almost five-sided. Almost looks like a square.
Yeah,
So that's
go
another
ahead.
piece of information to factor in into explaining the origin of the mound itself.
Yeah,
Oh,
I believe so.
So another tin can here,
or steel, to be accurate.
steel can. Thank you.
Little iron enrichment
Yep.
going on here.
Throws a label.
Some Serpula tube worms on the outside.
And coming out.
Okay, thanks.
Full wide.
Yeah, I agree, video. It looks like this stuff on top of the sediment is actually what's
Right.
decreasing visibility, not the actual.
I've
So
seen
another
that somewhere
word
else, but
for
I can't
iron
place it.
enrichment is rust. Fancy way to say rust.
You know, it's
A
more
rusty
like
can.
shallow inland waters. You see
Yeah.
that.
Maybe this is a good time to talk about iron as a limiting nutrient in pelagic waters.
Yeah, I could talk about that. So, especially in certain areas of the open ocean, iron can be limiting and can be limiting to certain algae that may need it.
What are the black
So
things, though?
that means that productivity can be low. So algae need the iron to help with their cellular processes. And without that limiting nutrient, they aren't able to grow and bloom, and thus energy can be limited. As in the ocean, most of the energy is created by phytoplankton. So they harvest the sunlight to create food for other organisms, and that moves up the trophic chain. The
Video,
other
you think
option
we could
is
make an observation on these
energy
darker
through
objects on the
chemosynthetic
seafloor?
activities.
Sure.
Seem very light.
But most of the
I'd
deep
like
sea
to
floor
find
where
one
there's
we
no
could just kinda
chemosynthetic
image. How about
Yes.
that?
areas,
maximum
the deep sea really relies
Yeah,
on
roger that. I don't know if that's
material and
plant
energy
material
raining down from
or
the surface.
Organic.
So there's certain areas of the ocean where iron
That.
is limiting, and there's been thoughts to enrich iron in various ways. But I think there's also some negative consequences that people aren't
It's light
thinking
'cause it
about
seems very susceptible to current
if they do enrich certain areas with
and
iron.
ROVs.
And that could lead to increased CO2 in certain areas, and we all know now that increasing CO2s in the ocean is leading to a lower pH and ocean acidification.
I
So
see a bunch there.
lots of different sides of that to think about.
The most obvious thing you can observe is that's why the open ocean is blue rather than green.
Yeah, absolutely. So with more stuff in the water, more algae in the water, you don't see quite a blue light. Blue light can penetrate deeper if the water is clearer.
Well, it looks like it's watch change time. So I'm going to
Getting a
hand
nice
off
view
to,
of all those different
Todd.
species of pteropods, of all the different tests of pteropods. So as I said before, there's
You
a
okay
few
with the
different
full zoom
taxonomic
for the
groups
handoff?
of pteropods.
Thank you.
They're swimming sea snails, sea slugs, sea butterflies, and they're in the mesopelagic water. And gosh, you can see quite a diversity of these mollusks now here in this little patch of sediment. We're at a depth of 2890. Thank you, team. Thank you, Dan and Ed. Pilots, co-pilot, Nav.
No problem, Andrew.
Great video work this morning.
Watch lead video.
Go ahead, video.
Trying to get some good observations for you of these longer dark matter that's on the seafloor. I don't have any in frame right now, but they seem very susceptible to current. There's some at the bottom frame there.
I think they're sargassum blades.
Yeah, that.
Yep.
I attribute their susceptibility to the current to being very light
Yes.
in weight and mass.
Whereas the pteropod shells you don't see moving,
right.
picking
No,
up,
I
moving
don't
with
see
the
those
current.
up. Seems to be what we're stirring up with the ROV, though. I assume once we get to the feature, that won't be an issue, but
Yeah, so it's interesting just thinking back to ocean acidification and, higher CO2, lower pH. Some folks are studying effects of ocean acidification on pteropods, on live pteropods. So they're collecting pteropods, running experiments, high temperature, low pH experiments on pteropods. And it's just interesting to think about if these organisms can be affected by ocean acidification in the surface waters, how is that going to impact life on the
Hey,
deep
Jeff,
sea
kill
floor?
the arm. Thanks.
Thank you, RV team.
Yeah, Santiago's noting there may be some urchin spines in here as well.
So Sirius is moving and starting to move past you.
All right,
There's something in the water column right there.
full zoom.
let's come out and
Nice
get
big
that fish.
I don't know if it's way up or sort of.
Ooh,
the
fish,
bottom.
big one.
Go chase it. I'll come up if I need to.
Okay, coming off bottom.
Thrusters all look good. Sirius thrusters are good. Sirius power's good.
Yeah, we're coming up a little bit. There was a fish sighting. I can see it in the Sirius view.
Down. You got plenty of tether.
Fish, fish, fish.
think it's
Sighting
up
right
a
at down
little.
to 2,888 meters.
Touche,
Trying
as they
to find
say.
it here. There he is.
There he is.
Yeah, I'll
He's
come up
way
to it.
I'm coming up.
Roger. Both vehicles coming up.
Nice view of D2 from Sirius. You're safe to get in there.
Okay, video. You want to start coming in?
Coming in,
Jason
I-
runs off. Can we get it from here?
Big cuskeel. Ooh,
Go get it. Go get it, Jeff.
Get him, get him, get him.
make him turn, make him turn. Looks like
that's a
some parasites on the
close to
side, too.
That's a great picture.
Oh, it's moving pretty quickly away from us. Definitely tell it's an
Just keeping him in
ophidiid.
frame with zoom. Let me know if that's throwing
It's
you
got
off.
pelvic
You're
fins that
okay.
are long and filamentous.
He's getting away slowly.
I think he's dropping. I'm going to go
Yeah,
down
he's
with him.
dropping.
Okay,
I-
thanks, Jeff.
No, that's okay. I got him still. I just,
All
I'm
right.
Full
I gotta-
wide now.
I'm going to come down. He's getting closer to the bottom.
Hot pursuit.
Just trying to get a side view for you.
Oh, there you go. Get slight zoom.
Oh, he's turning now. That's great.
Actually pirouetted a little for you.
Yeah, and it looks like there's some nascent isopods on this side of the fish.
Is the drawer closed? That ball's in
Yeah,
the way.
the drawer's
Let me take
closed.
care of that.
Full
Slide
wide.
straight down. Thanks, video.
Starboard close. There, that's a little better. You like that?
Yeah. Just trying to come down to him.
We're on a
here.
fish chase right now. Try to get a better view.
This bottom stirs up very easily.
I'm getting.
The material on top of the bottom, not as much as the bottom.
I'm looking orthogonal to track,
Copy.
and you're getting out at the end of the tether. I'm about to feel a tug here. Ready, tug. That's it.
alright.
Done. Game over.
Snap zoom. Go for it. Video right here.
Roger,
Actually, this
right
could be
here.
a
I'm going to
bite
spin right,
fitted.
give you a little more tether,
That's all you got.
just for a second, and then you got to come catch me.
There you go.
Yeah, it's several parasites on this side. One on top of its
I'm
head, a
pulling
couple
out.
on the side.
Yep. And that's it. Let's go.
Great,
Roger
thank
that.
you.
Full wide. Both
255
cameras.
is the move.
You're pulling me. Just walk it over. Don't turn over.
Sorry.
watching fish eye.
Thanks. Still coming.
Nav, what's our move? 255.
255.
Video snap
Okay.
real quick. See what this
What's
is.
this current ship speed?
Right there.
Point 2.
Not a
Point two.
fish.
Thank
Okay.
And
Come
the
back
first
up.
Holothurian of the day,
Thanks.
sea cucumber.
On the ship.
It'll come off the bottom and put on a swimming show for us. Nope, it's going to stay on the bottom. And we're moving
Think that would
quickly
actually just
towards
keep
the
ship
seamount.
moving.
I ran off pretty far to my port to chase that fish, so I'm just
Probably.
coming back
We
over
got a long
towards
way to go
Sirius.
uphill, and there's
Whoa.
you're kind of below me right now, Jeff.
Copy.
I think you're dead below me.
Great.
Try to tilt down and see if I see you.
Coming in to view soon, hopefully.
There's your light pool. There you are.
Sweet.
I'm going to spin right and get on track.
So Jeff, you're alright if I get this move?
Dog club.
Sorry, video. We're just that fish chase got us pretty far off track, so we're
Just calling them
got a Holothurian track on
to your
too.
discretion.
Okay. Sirius is pretty close to being on track, heading-wise.
Copy
Right
that.
now, from under me. Tilting up, coming up.
Got rock. Did a hit. Pilot video, shift change.
Thank you, video.
Thank you.
Pilot Nav, thinking about extending this move, which is the ship's just coming to the end of a 100-meter move.
Okay.
So just keep it going.
Just a couple rocks here on sonar coming in.
Yep. A few things interesting to look at, but
Our first rocks of the day.
keep pushing
Great.
on.
Big walls.
Yeah, there's
Nothing on
Possibly
Sirius
a
view.
few sponges colonizing small ones on the side. It's
Bridge
very
nav.
scoured around. Current scoured.
Yeah, this
We're
is
at a
bridge.
depth of 2,881
yeah, it looks like
meters.
we're coming to the end of the move. I'd like to extend that target by another 50 meters, please.
Copy that. Another 15 meters. 255.
I've got a
Thank you.
squat lobster possibly over
Really?
there. Yeah,
All right.
hold on.
Look at it.
Settle in. Video's doing a watch change, so we're just gonna just get close first, and then when they're ready, we'll zoom in.
Okay, we have a squat lobster, several different sponges growing on the underside of this rock. Again, the top is covered
Very
with a
easy
lot
to
of
kick
sediment.
up the top of this sediment. Very easy. Video, come on in on the squat lobster here.
Just so you know, I'm making steady progress over you and uphill.
Yeah.
Oh, again, this white
light layer of
morphotype
sediment
of the
on top, look at
Munidopsis.
that.
We have seen this at similar depths in the Mona Canyon, I believe. And possibly the first dive on the Arecibo
All right.
Amphitheater.
Video. I think we're not going to get a beauty shot of this.
It's kind of
so
got this really pointed
That's enough.
rostrum. Points up, curves up.
jump up over this rock and move on. Learned our lesson. Go a little slower.
Looking straight down at you.
Yep. Just picking up.
It's interesting that
Over
the
this, so
sediment
I can
is a
push
lot
ahead.
more fluffy here. It's lighter. It seems to be stirred up.
It's
A lot
very
easier than it was just a little bit ago.
Yeah, indeed. We're seeing a lot more, when the ROV gets close to the seafloor, a lot more disturbance. Looking at the rocks again, we're seeing this ubiquitous iron manganese coating, which makes it hard to ID the rocks. And we're just peering to hopefully figure out what they might be.
Tilt up, try to get you out there.
A few more up ahead.
The rock ID could be very difficult today. We really are not very sure what the rocks are down here. They could be the limestones again. They could be the Cretaceous volcanics that lie below the limestones. And there's even the chance that they could be some more recent basalt, but that I think is unlikely. But we really are on a voyage of discovery in terms of the geology today.
Can you explain to our viewers what that means, more recent basalt? To the east of us, of course, is a big volcanic arc, and the Lesser Antilles arc, where there's some big volcanoes like Montserrat, etc. At some point, that volcanism extends west. There is just the chance. I think it's very unlikely that some of the rocks in this area were volcanic, related to the rifting, maybe related to that arc. But it's much more likely if they are volcanic, they are the Cretaceous to Eocene basement to Puerto Rico and the Virgin Islands. I think there's a fish up ahead, Jeff.
Fish up ahead, upper left. See it.
Looks like a lizard fish, maybe.
See it in Sirius HD as well.
Pilot, our light pool looks
So again,
a
we
little
see
shallow.
some sponges colonizing these rocks, but they're very small, not very abundant.
Okay.
It's steep slope, so.
Copy that.
Do you want them swung down? Or?
No, the opposite.
And
it looks
you
like
can see this shrimp on the left with long
was
antennae
falling off
moving its
pretty sharply, but
pteropods
that
or
would be explained
swimmerets,
by
You want to come in
kind of
from
paddling in the water. Yeah,
Get a
so we have a
quick view.
Bathysaurus,
And
deep sea lizardfish.
a small one.
Yeah, it's not very big.
I get the lasers.
Lasers on. Getting behind
So
me
these
again.
are primary deep sea fishes. Don't know a whole lot about their biology, but we can tell from the way that they're
we're just gonna get
positioned,
the
their
size
mouths,
here,
the
and then
size
I'm
of their
gonna move
teeth.
on.
They're likely piscivores and crustacean eaters as well.
Up with Sirius. All right.
They're mostly found below 1,000 meters depth, so that's Bathysaurus mollis.
So Jeff, again, in terms of the geology, what we're kind of looking for is if you ever see a rock that has that black stuff knocked off it, we can see into the inside of the rock. All the black stuff, of course, is the iron manganese coating. So if you're flying over and you see a lighter face, or that would be great if we could go there. I mean, overall, what we're seeing is what we've seen many times at the bottom of slopes. We're seeing the talus pile that forms at the bottom of the steeper slopes. These are almost certainly all boulders that have rolled down the hill. And we can see little sediment chutes. You can see that coarser grain gravelly material running down the center of the picture. You can just imagine this stuff breaking up and rolling downhill, maybe being shaken every time there's a big earthquake. And now we're coming to a much, much bigger, beautiful, serious view where we can see extensive outcrop. This may well be in situ.
Are the upper light bars on?
Where we need to start looking to see if we can ID the rocks.
Sure, yeah, they.
Nav, actually.
Go ahead, Watchley.
What is the length of our ship move right now?
We're on a 50-meter move, which we're maybe halfway through. That's the ship. This depth, that means that Sirius has quite a ways to go.
Okay, we may want to just do smaller moves, just to move a little slower.
Okay.
Yeah.
We're cruising.
But we're good right now.
If we've got time, if we can just zoom in close to...
Watchley, do you think we slow down?
Try to push out when you can, Jeff.
I think right now we're okay, but we may need to slow down.
Yeah, I mean, slowing down will have more of an effect. If we
wanna
keep
go
making
like
shorter
partial
moves,
video
it
for
doesn't
some of this,
matter.
we can. Just
Okay.
But we could speed.
Just get a good look at it.
The
That's good.
DP system only knows about
That's why I like
tenths
this.
of a knot. Right now we're at
Closer
2/10.
view of some of this stuff
We could
while
slow
go
down to
by.
1/10.
We could slow down to 1/10. That would be great.
Yeah.
Thanks.
So again, again, in terms of rocks, you can see they're black. This is this iron manganese coating that makes it so hard.
this bridge.
Yeah, we'd like to slow our
see
speed
this
down
sort
to
of
0.1 knots.
knobbly character.
Do you want me to stop the position move and then adjust it? We've only got about
One of the things I'm
10
looking
meters to
for
go.
is with the limestones,
no,
you
that's
see this
okay.
characteristic pitting,
Okay,
this
every other move will
honeycomb
be
texture.
0.1 knots.
And I'm
then.
not sure we're seeing
Okay,
it
so
here.
you can't change it in the middle
I
of the
think the texture of these
I could
rocks is
try.
slightly different.
Not quite sure what the response would be.
but it is
Okay,
very
fine.
difficult.
Thank you.
All right, video, come on out.
Thank you, Chris.
Coming up, pilot.
We're at a depth of 2,843 meters. This is the 11th dive of the expedition to survey deep waters, unknown and poorly known areas around Puerto Rico and the U.S. Virgin Islands. Currently, we are 20 miles northeast of St. Croix, surveying Exocet Seamount in the Alligator Passage. Moving upslope.
Again, we can see the fine sediment coating the mud on the left that slowly builds up and covers these rocks. But we really are in massive outcrop. There's a beautiful view in Sirius.
Yeah, just going to comment on that.
And,
Co-pilot,
and a hint of this thing we talk about a lot, bedding. If you look in the Sirius picture, you can see a kind of orientation from the northwest to the southeast.
Quite a good while,
and
probably 10 or 15 minutes till we...
Bedding is something that's characteristic of sedimentary rocks.
Just pause for 5 or 10. We're really cruising.
Okay. Yeah.
Just steady up on the winch.
Can we zoom on the... There's an anemone below you, Jeff, or is it too late?
What do you think, co-pilot?
Quick.
All right, I'm going to hop down here.
Can do a quick snap zoom is fine.
The anemone is...
Keep going. Yep, right there on the left.
On the left. Oh, I see it.
Yep.
Jeez. How did I miss that? I don't know.
We're moving pretty quickly.
All right, video, come on in.
Okay, thank you. A beautiful Cereanthid tube anemone. So we're seeing all the tentacles, and they actually can retract down into a tube that they make from aggregating sediments and different things to make these long tubes that they sit in.
Continuing up.
Nav watch lead.
Yeah, go ahead, watch.
Were we able to slow down the ship?
Yeah, I believe the ship is slowed to point 1, and we'll be reaching the end of the move very soon.
Okay, great.
It'll take,
yep.
We're at a depth such that it takes 15-20 minutes.
Bucket, not a rock of different color.
Another steel can.
It takes quite a while for Sirius to realize the ship has stopped.
Yep, totally understand.
So what I was discussing with pilot and co-pilot was just letting things settle out here for maybe then 10 minutes or so. Things will slow down and then move on at point 1.
Sounds good. Thank you.
So it really does look like a slope with all this rubble, these boulders of different sizes, some meter scale, some 5-10 centimeters, probably smaller, just sitting on outcrop. And I'm going to guess, and it is really a guess, that this is the Cretaceous Basement. And if it is, this is the material that sits underneath everything of the islands in this area. It's the core of Puerto Rico, it's the core of St. Croix, it's the core of the rest of the Virgin Islands. And as I was mentioning earlier, this whole area is really like a cork that's being squashed by the big North American plate to the north and the Caribbean plate to the south. And so this is what's in the middle of that cork. Chris, if you get a chat, there's a little bit of biology. Is it a sponge? Can we zoom in on that and see the rock at the same time, center of the screen?
Thank you. There's a Munidopsis sweat lobster on the left.
Yeah.
And I just have a request shoreside. I'm going to go check and see where we are with getting the nav feed into the event log. But if you join the nav data chat room, you can enter the lat long, depth, temp, salinity, all that. You can find it there. And once in a while, if you could copy it and paste it into the event log, that would be a huge help to us, especially as we get kicked out frequently. But I'm going to go check on that right now. Thanks.
Thank you, Amanda.
Looks like the ship has stopped.
Yep.
Okay, video. Want to come in on this?
Okay, indeed, we've got a little sponge, and Art is still taking photographs. So again, you can just see the sediment dusting on the iron manganese coated rocks, and we're just looking for clues about the rocks, whilst also looking at this beautiful sponge. And I'm guessing it's a glass sponge.
All right, if you want to track focus to the rocks for geology. Great. Good view of
So in terms of the geology, guys, we're done. I'm just looking for clues. And I guess, back to what I said before, it doesn't quite look like the carbonate.
Zoom in closely, we see several more small sponges here. That glass sponge, hexactinella sponge, is pretty cool. See the long spicules sticking out. Spicules are structures that are embedded in the sponge tissue. Also provide some support, protection, and are useful for identification, just similar to octocorals, the sclerites found in octocorals.
All right, video.
It's good for me. Is that good for you?
Yeah, thank you, Chris. Thank you, video. Thank you, Nav.
That's Jeff we have now.
Sorry, Jeff.
That's okay.
Us
the
all,
ROV pilots all answer to any of our names. We get it.
That's just us old guys. We get things mixed up.
I'm flattered you think my flying is good as Chris's. So.
You guys are all great.
So can we pan back right?
Oh yeah.
Quickly,
I thought you wanted to see that lobster. Sorry.
Well,
lobster.
There's just a mention in the chat room. Sorry about that.
That's okay. Just going where I.
It was just came through.
It's not a problem.
There was something stocked on top of the rock where we were. I didn't see it. Amanda, are we on track?
Try to get a little closer to light it up. You'll see it. Oh, I think I got it.
You do?
Yeah, stand by. It's coming into center screen now. It's kind of waving.
Oh, okay.
Come in a little bit.
It's like a stocked sponge of some type.
Is this what we're talking about? It's the only stocked thing I see so far.
Stocked item was the top of the rock where we were earlier.
Oh, okay. Come back out, video.
But those are some interesting sponges.
There's a couple there.
Yeah.
Pushing up, looking for a stocked item.
I think was it below us?
Well, running out of time.
Yeah.
To keep looking here. But I could look below us briefly.
Another Munidopsis. I'm not sure where we were.
I'm not seeing it.
Okay, let's
Sorry.
continue on. No, that's okay. Came in too late.
Jeff, my track is to the right of my head, but I've started to stabilize here.
So two
Not sure too
new,
much.
two squat lobsters.
So again, just this hint that there might be bedding from the top right to the bottom left. You can always kind of draw a line. You may be seeing layers in these rocks.
So it looks like our nav feed is back up running into the chat room. So thanks for working on that. The depth right now is 2,810 meters, temperature 4.0 degrees Celsius, dissolved oxygen 7.5 milligrams per liter. So Amanda asks, what is our range to the rocks in front of us? How far off?
About 3 meters, maybe.
3 meters, okay, thank you.
I'll get in a little closer when we're not, when we start feeling that slower move.
Yeah, it looks good right now.
Jeff, take your time.
So Jeff, thanks.
The faster you move, the more cushion you give yourself to react to bumpy rocks.
You know, it's interesting. So yesterday, I'm just thinking about yesterday's dive, and the rocks that we observed had manganese coating. Again, we were in much shallower depths, around 800 meters, but in terms of sediment, is the sediment draping here look any different from what we saw yesterday?
I think less. I mean, we're seeing a lot more rock.
Yeah.
And whether that's a function of things like the currents coming through, or whether it's just slope. But I do think that these rocks have a different character. And again, it is so difficult, but the more we look, the more we ride around this slope, the more I can imagine that it's this low-grade metamorphosed volcanic terrain that forms the core of the island. Nothing is honeycombed. We're not seeing
moving very, very slowly.
anything that looks like it could be a sequence of sedimentary rocks. My best guess would be that we are on the core of the plate. We're in the Cretaceous to Eocene volcanic series.
Nav.
We're really not seeing any boring or anything like that.
Exactly.
Pitting in the rocks for
Nav this bridge.
Exactly.
Yeah, we'd like to do another
bioerosion.
move, please. 50 meters at 255, 0.1 knots.
50 meters, 255, 0.1. Copy.
Thank you.
There's a holothurian coming in the top right.
Another glass sponge right behind it as well. Can we get a zoom on the glass sponge behind the holothurian, Jeff, please?
Yep.
Looks like something a little different.
I'm going to come down a bit. I'm giving you the slightest little bit of tugs.
Copy. I plant my toe here. Alright, video, come on in. Hold. Adjusting my head in.
More interested in what's on the left.
On the left?
You see that? Is that
Another
another
sea cucumber?
holothurian? Yeah. Looks really different than
All right.
others that we have seen.
It's within the realm of possibilities. All right, video coming in on that. As I bring it into the center.
Wow. Very cool. Definitely different.
Full zoom when you're ready. There it is.
Full.
Thank you.
So we're just getting an image of a different white holothurian. I do not believe we have seen this yet on this expedition.
The holothurian in our minif cam, Todd. It's not like it's just copy pasted there. Doesn't even look like it fits the scene.
I know.
And
right?
holothurians are sea cucumbers. They're very diverse in the deep sea. They are deposit feeders, often selecting organic content out of the sediment, filtering it out. And some people have asked whether we've seen any associates with the sea cucumbers. We know in shallow water sea cucumbers there's often pearl fishes in the family Carapidae that actually goes up the anus of sea cucumbers and occupies the respiratory tract. But we have not seen any associates with deep sea holothurians.
Okay.
To my knowledge, anyways. Thank you, guys. See if somebody shoreside knows the identification of that. But we've been kicked out of the chat room. We're at a depth of 2,799 meters, surveying Exocet Seamount.
So again, we're carefully looking at the rock surfaces, try and see if we can find a piece of rock that's recently broken, so we can see the interior of the rock beneath this iron manganese coating, and that will give us a better clue to what we might be looking at. We are in a very cool part of this area. We're in the Alligator Passage, which is made by the edge of the plate. This is the edge of the Puerto Rican U.S. Virgin Island microplate, and the tectonic activity there, the big faulting, has created an enormous rift-type structure, and people argue on how it formed, whether it formed by slippage to the left or slippage to the right or east-west extension, but it's exposed all these rocks, and each of the seamounts is essentially a piece of the geology that's been left standing in the air by this pulling apart, and the seamounts are the bits that are left sticking up after the crust has been pulled apart.
And I just want to add to that, this passage again is where a lot of the water from the Atlantic flows into the Caribbean at deeper depths than, say, the Mona Passage. The sill depth is about 1,900 meters, I believe, in the Alligator Passage, so the North Atlantic deep water can flow through here and enter the Caribbean Sea. The summit of this seamount, I believe, is one of the more deeper summits than the other seamounts in the area, like Norwa and Dog and Conrad for sure.
Yeah, Amanda and Jason from the USGS. We're on a cruise with the Nautilus last year, I think, that looked at the seamounts to the northwest of us. And this is one in a series of 4 or 5 of these seamounts. This was first looked at geologically by Genet in, I think, 1990. And he concluded from the magnetics that it might indeed have a volcanic basement. And that's looking like what it might be, but there's very much this Cretaceous volcanic basement. I was talking on the event log with Amanda earlier about what the geology was of the other seamounts. And those guys managed to sample. And again, it still remains tricky, but it does look like
that's something to look at.
They were volcanic.
Biology-wise.
Yeah, can we get a zoom on that from the Sirius view?
Well, there is a fish in the Sirius view. There's also another one of those.
Is it possible, co-pilot?
Co-pilot, do you copy watch lead?
Oh, sorry.
Sorry. Come back.
If you tilt down a little, I think there's a fish there. If you wanted to try to catch a zoom. I think like a rat-tailed fish.
I think it was a cuskeel or a snailfish.
Oh, okay. Well then, all the more reason to zoom.
Go in, video. Feel free. I'll try to center it up as best I can. Could be. Dip down, Jeff.
Kind of a hard shot here. We're at a depth of 2,792. We're just zooming in with Sirius. There's a fish in the Sirius view.
Come out a little bit.
It's hard to tell if it's a cuskeel or a liparid.
It's swimming away from me.
Thanks, guys.
Spin back.
There's also, I think, an anemone here.
Okay, great.
It's like the one we took a snap on earlier.
The tube anemone. Oh, I see it now.
They blend in with the rocks
pretty
well.
Well spotted, guys.
So we're getting a zoom in on what looks to be a
video.
Serianthid tube anemone.
If I can here.
Really long tentacles. So another cnidarian. On our descent, we saw a few jellyfish. So jellyfish are related to these Serianthids, all in the phylum Cnidaria, meaning that they have cnidocytes. Particularly, many of them have nematocysts or stinging cells.
Just trying to get perched so it's centered here. It's not a lots of little bumpy rocks, not one good one to perch on. But you can come in a little farther if you want video. Take some of that sediment out of the view.
And then getting a good view of the mouth, oral part, the center of the anemone. Yep, and you can see the inner ring of tentacles surrounding the mouth It's a really good diagnostic character of the cereanthids. Pretty
I'll hold
color.
here for a minute, Pilot.
Spectacular, guys.
Good morning, Chris Kelly joining us from Hawaii. Just commenting that they have this species, possibly, definitely this morphotype off of Hawaii. Beautiful.
I'm ready when you are.
All set, thanks.
Lifting off. Pushing ahead.
So coming to the end of this move, should I keep it moving at a 10th?
How you feeling, co-pilot?
All right, continuing to 791 meters.
Good. I'm looking pretty much right on track.
Super.
And we're starting to see some quite steep topography. Again, a superb view in Sirius of this as we wander around this outcrop. Again, the hunt is on for the broken face that will give us a clue to what these rocks are made of.
So far, this dive is comparable to what I have viewed on the Seamounts. That's similar. We haven't been this deep. Let me look.
Bridge nav.
So yeah, we haven't been this deep yet on Conrad Norrois or Dog Seamount. But still, towards the base of the Seamounts, we really didn't observe a lot of attached fauna.
Bridge nav.
More as we got closer to the summits, did we pick up a lot more sponges and corals,
Now this bridge.
particularly at the
Yeah, we'd like to add
shallower
another 50
summited
meters to
seamounts.
this move, please.
Hi,
Adding
Susan.
another
Thanks
50.
for joining
Thank
us. We've been on bottom now for about 45 minutes or so.
We hit bottom at 1328.
Have observed so far 3 different species of fishes, the deep sea lizardfish and 2 different cusk-eels. A few sponges, but not a whole lot of colonization of attached fauna on any of these rocks. Can we get a snap zoom of the sponge coming up on the rock?
Stocked one?
Yep. Thanks.
Absolutely. Just a quick one, or you want a good view?
Yeah, just a quick one's fine.
Right,
I think
VA,
we've seen this
coming in from
before.
here.
Just need for an ID.
Good there.
Beautiful. So we have this stalked hexactinellid glass sponge. I know we have seen this on previous dives. Hopefully we'll get an ID from shoreside here in a minute. Looks like a tulip. I think that may be the common name of it, a tulip glass sponge. Thanks, guys. That's great.
Turning the Z-bias down a little. I think it's pushing me down generally. Thanks, video. Come wide.
So we're now traversing over a more muddy part of the area. We can still see the talus boulders poking through, but there's more mud cover.
So Susan asks if we start at the bottom of a talus pile, or has it been like this the whole time? It's been like this the whole time, right?
I'd say it's changed slightly.
Oh, has it?
We started on a talus pile, and then we got to perhaps a steeper slope where there was much more rock compared to the mud.
Okay.
And so there, I think we were maybe seeing what's underneath the talus pile.
Cool.
And then I think now we've come up, and my guess would be that, this is partly a function of steepness of slope, is we've now come up and we're in a slightly shallower part of the slope, and so the mud is more burying the rocks that are underneath.
So, to my geology ignorance, what exactly is a talus pile?
Talus pile is just a name for a pile of loose rocks that build up at the bottom of a slope. Another word would be scree. So talus or scree are the individual rocks, and we've got a pile of those rocks. We can see them if we go and look at the continents.
Thank you.
Jelly?
Yeah, let me slow down. He's tracking towards me. Okay, start coming in. Hold there.
So tiny little jellyfish.
Okay, come on in.
Trachymedusa, perhaps? We have observed this jelly a few times during this expedition, but this one's pretty small.
Yeah, that's nice. I'm glad I didn't leave yet. I think that's small hydromedusae. I think it looks like possibly from Crotata.
Could you repeat that one more time, hydromedusae?
I think this one, looking quickly, might be from the genus Crotata.
Crotata. Oh, I moved pretty quickly.
Or maybe this is more like a Epicodon genus. I'll correct myself on that. Pretty nice. The tentacle results here in the pictures are excellent. And like so many of the deep water jellyfish that we see, they have a pretty serious pigmentation.
Again, this red color that helps these animals be camouflaged in areas where there's light. As red is the first color that is absorbed in the surface of the ocean.
All right, I'll let them float off.
So, Mike, can you repeat? Did you change the genus name on that?
Yeah, sorry. I'm gonna call that Benthocodon.
Benthocodon. Great, thank you.
Yeah, that would be the genus, and it's a Trachymedusae.
He's so small. Look at him.
A Hydromedusae.
A Trachymedusae is part of the Hydromedusae, is that correct? Are they two different groups?
If you look at this...
No. Okay. Trachymedusae is within Hydromedusae. Sorry about that.
Yep, that's why, okay. I got a little confused there for a minute, but okay, thank you.
While we zoom in on our next biology and a shrimp going by, if you look at the Sirius view, it's kind of cool. You can almost see a roadway in the mud and the rocks. The rocks are forming, oriented in a kind of straight line from the top to the bottom left. Again, that might be revealing some structure within the seamount.
Great, we're getting a really good shot of the shrimp, really long antennae. You can see the pleopods moving, the swimmerettes, like paddles moving the shrimp. And again, these long antennae kind of folding back around the body are really good for detecting any movement and possible predators coming up from behind. My guess is the ones out front are to detect prey.
All right, video. Getting behind a little, so I'm going to catch up.
Some more sponges on this rock block on the right.
Science, when we see those shrimps and crustaceans kind of do a tail flip and shoot backwards, is there a word for that?
Not that I am aware of, but I can ask shoreside.
Yeah, I seem to recall it's a carioid or cardioid response. Fairly common that I did not have the correct name, but something.
Let's see if
close
anybody
to.
back on. Oh, yep, Scott knows it. Carioid response. Yeah, good job.
Thank you.
The Sirius view is really quite remarkable. It really does look like we're driving up a mud driveway between rows of rocks on our left and right. Jeff, if you can get a chance to kind of snap zoom close in on one of the rocks, your choice. Again, just trying to look at the texture of these things.
Yeah, okay.
And that's kind of cool.
Yeah,
if
you
you look in the D2 view, you can see the layers at the top. Maybe we found bedding. Thanks, Jeff. That's great. Can we pan up? Remember when we came down, there was a layer.
Up, sorry.
So, yeah, zoom out and pan up.
Yep. Okay. Video, could you come out?
And so now coming into view, can you see the layer at the top of the screen? Can again we...
Am I getting close here?
Yeah. Okay. If we could zoom in, maybe?
Yeah, I'm just going to get the vehicle a little closer. We'll be able to light it up.
Yeah, sorry, thank you. It just looks like they're actually just blocks sitting there.
So there's another hexactinella tulip sponge sighting, the Hyalonema species. So Scott France definitely confirmed that tail flip of the shrimp we observed, called a caridoid response, but it's named for the type of shrimp, the Caridian shrimp. Must be a common behavior in the group. Thanks, Scott, for that.
Thank you, guys.
So again, just really small sponges here.
Right.
Oh, is that a coral on the right?
Looks like a little coral.
Or an anemone. Can we get a close zoom on the...
Got any more video?
Yeah, looks like a cup coral. Yep, sure is. First coral of the day. Here we have a solitary cup coral. Yesterday we observed several colonial corals such as madrepora and analopsamia. So this is just one polyp coming out. All the tentacles are extending out to feed, and you can see inside these septae, these kind of linear structures surrounding the calyx of this cup coral. again, these are used diagnostic characters
Cephals,
for
yeah.
identification.
Full.
But cup corals are hard to identify with just photos alone. Often we need specimens to get appropriate IDs. It's a beautiful image. It looks like there's a hydroid to the right of this too, so another Cnidarian.
There's one above it, I think, as well. Maybe. A couple few.
Yeah, and a couple above.
Okay, video, I'm ready when you are.
Thank you, ROV team. That was great. Again, I'm fascinated by the Sirius view. It really looks like we're in a roadway with outcrop on either side.
Yeah.
Full what?
And we should figure out the orientation of that feature as we come around.
Yeah, like a dirt road in the backcountry
Yeah,
or
it's
something.
And so the feature's just about east-west. Look at that. That's amazing. Great view out of D2. You're just seeing along that outcrop. It's almost as though that there's a hole, if you like, a channel been weathered down through the middle of the outcrop.
Is that a small red shrimp in the bottom of the screen?
You want that?
Can we zoom shrimp?
Zoom shrimp.
Looks something to be something different.
Track in video as I come in.
Beautiful. Again, this common color that we see in the deep sea, this bright red. Very long antennae, long legs.
Yeah, this is also a Caridian shrimp. It's in the Nematocarcinidae family. It's a family in that genus actually is found throughout the ocean.
Could you repeat that? The genus what?
Very thin legs.
It's in the family Nematocarcinidae. It could be in the genus Nematocarcinus.
Nematocarcinus. Great. Thanks, Tim. And we got an ID from Cheryl Morrison earlier on that cup coral to be Desmophyllum. So thanks, Cheryl, for that.
Andrew, there's something white coming off the front. Oh, no, I'm sorry. It's just it's like cilia coming off of the antennae.
Oh, yeah.
See the base of the antennae coming off the head?
Yep.
You see white material. I wasn't sure what that was.
So could you tell us a little bit more about that, what that structure is for?
Well, it's to trap particles that are coming by. It looks like these antennae, obviously, are for filling the seafloor. And it does very well on sediments, doing those long legs. But those cilia probably collecting particles that are coming in the water column. Didn't really have a great view of them, but there are numerous implants that have those. I don't think Nematocarcinus does, so they have to reevaluate the genus on that.
Okay, great. Thank you, Tim.
Yep.
Okie dokie.
Bridge nav.
And we're at a depth of 2,765 meters. Just want to stop for a moment to say thank you for our shoreside support, including Amanda Demopoulos, Bernie Ball, Cheryl Morrison, Chris Kelly, Randy Singer, Santiago Herrera, Scott France, Thomas Ritter, and Tim Shank, and Mike Ford. Thank you guys so much.
Go, Nav.
Yeah, we'd like to add another 50 meters, please.
Copy, another 50.
So this ledge that we saw is now less pronounced, but it still continues, trending east-west. The ROV is just facing a little bit south of west, but you can see it in the Sirius view, and as we travel along it with D2.
Snap real quick video.
Maybe it's an indication of sedimentary layering, in which case we're above the volcanics.
Hold there.
Interesting fish. I'm not quite sure what this is yet. It's got a tail like a cusk eel. Can you get a shot of its head any closer, or is that full zoom?
It's very small, and we are zoomed in.
Okay, thanks.
Not quite full, but
you still see
I've
the
got a
lizards.
little more to go, but
a little
yeah.
more. So hold on. When he comes into view, snap in. We'll try to get it. Got it pretty so.
We have not observed this fish yet during this expedition.
Turn around.
Went vertical to horizontal.
All right, go ahead in. Oops.
You see one long continuous dorsal. And of course we get this end of the fish.
This is
Small pectorals. That dorsal starts really
Keep
close
tilting down.
to the top of the head.
His head looks
Wonderful.
damaged.
See. Oh, there's his eyes. Don't see any parasites. Can't really see the pelvics yet. But some type of cusk eel, or new for me. Haven't seen this one yet. Yeah, I agree. It's most likely an ophidid cusk eel. I think it's always, really fun when we see things we don't know right off the top of our head. We may be able to get them to a certain order or family, but beyond that, it's difficult. So we're still learning every day, all of us out here. Working together, it's been tremendously helpful. I mean, my knowledge has increased so much since we started these telepresence cruises. Being able to work with up to 40 scientists on shore at once, even when I'm on shore side and people are on the ship, the amount of information exchange that occurs is fantastic. So it's a really good learning experience. I know several grad students are watching, and undergraduates are watching and following along and tweeting, and they're learning their diversity, taxonomic diversity as well.
Yeah.
So anyways, when we see something we don't know, we can hopefully then review the video, send some photos to other experts that work in certain groups, and come back with hopefully an ID. Ooh, another fish, right?
Ooh.
12 o'clock.
Oh yeah, we have seen this already. Great, I got a close look at it. It's another cusk eel. There's two possibilities for genera, but we would need to get a good view of the side of the head.
Down.
Let me know when you're ready for zoom.
By 1 second. Just get a little lower.
So again, we're seeing boulders in this muddy slope as we zoom in on the fish.
Right there, he's turning their back to us.
And carefully trying to look at the textures of the rocks, these boulders, to give us a clue.
Scampering away. So we have observed this, I want to say, during the septentrional fault dive.
It might be. I remember
maybe the West Mona Canyon wall.
Maybe Mona Canyon.
Saw it twice.
Yeah. We have some beautiful images.
So this fish has some really pronounced spines that are coming off of its head or opercle. So it'd be good to get a good view. About 20 centimeters in total length. So you can see the one pronounced spine coming off its opercle or the gill flap. And I think there's another one above the pectoral fin.
Tilt
Kinda
up.
hard to tell.
Coming back. It's
Really bulbous head and pretty thin body.
It's acting unpredictably. Keeps changing direction on me. There we go.
Giving you some chase.
Go ahead, video.
You can see it's two filamentous pelvic rays that are actually right underneath the chin. They're really high up on the head of the body. Yep.
That was a good
Great.
shot.
Great job, pilot.
Thank you.
Coming
And
out.
video. Great job.
Pushing on. Got a little behind chasing around after that guy, so we're going to do a little jump here. It's nice to get out of the mess I made anyways, chasing around.
So the possible ID on this Cuskio, I'm hesitant to say it, but it's Xylasiba Meyersi. That's it. Xylasiba Meyersi. That's a tough one. I wish I took a Latin class. It would help. Also helps when you know the origin of certain words. Like a lot of names of animal species names are named after people. So if you know that person's last name, that helps. So
In that case, you might guess Myers.
maybe so. My favorite fish name is Xenolepidictes. All right, I'm being a little bit of a nerd back here.
I won't try and repeat that. So we're passing over these big rounded boulders. You can see in the serious view, it gives you a kind of scale for how big these things are. They're meter scale, some smaller ones in between. And again, just looking for the elusive clue of what these might be. What we do know is they're all covered by this ubiquitous iron manganese coating, which basically forms on rocks pretty much anywhere in the seafloor. And so if you're a geologist, you've got two enemies. The mud covers up the rocks and the iron manganese coating. And so you really have to go to areas that are reasonably active to actually see the fresh surfaces of the rocks.
Ooh, what's this? Another Trachymedusa, perhaps?
Sorry, go ahead, video.
Oh, no, it's not a jellyfish. Very cool. Almost resembles what we saw, the sunburst
Indeed.
creature.
But with a bigger central area.
Yeah, the central area of that other was very different. That one was white. This is deep dark red. Oh, great. Chris Kelly has an ID. Crosata species. Oh, this is a jelly. I'm seeing it now. So you can see the bell, the red color, and then all the tentacles coming off the bottom of the bell. This is very unlike what we saw earlier.
Still spectacular.
Beautiful.
Yep, I see nicely rotates around.
So you can see the insides really well, the gonads kind of on all those canals moving along the bell. Great day for jellyfish. And Santiago Herrera is just noting that they also saw one in another area, the Celebes Sea.
Let it float out, Pilot. That was good, thanks.
Which, if people don't know where that is in an Indonesia area, in the western Pacific Ocean.
First large sponge colony here.
Oh yeah.
Yeah.
What a good view of that. We have seen this before on previous dives. Another Munidopsis, the white morph type. Again, common at this depth, a little deeper.
So, Andrea, any particular reason for why you suddenly got this largest sponge colony just under that one particular rock?
Yeah, it's a great question. I mean, it's obviously done really well. Found a good spot to settle and grow and survive.
Yeah.
That's the most important part, is surviving to adulthood, surviving past the larval and juvenile stage.
Video.
A lot of small glass sponges at the top as well. Going in for a better view of the sponge, see if there's any associates with it.
Oh, there is. Top left has a little red.
So the white portions that you're seeing is the live tissue. This is an animal. This is a sponge, and the phylum Porphyra. Oh, that's a swimming polychaete right there, I believe. Wow, beautiful. I believe the genus of this is called Swimma.
Swimma.
I think.
Back to the sponge.
Yep.
And there's a small shrimp right there on the sponge, shrimp associate. So the live tissue is the white, and then the darker brown is portions of dead tissue. It looks like there's also some sediment covering it. Can we pan up to the shrimp? Great.
Bridge nav.
Tim, are you still online?
Ready when you are, video full.
Bridge nav.
Ooh, exciting, Chris Kelly. Thank you. Not really sure about this genus. It could be in the family Uncinataridae.
Bridge nav.
Tim, are you still there? It's a gravid holding eggs underneath.
Andrew, yeah, I'm here. I'm just right over here looking at it now. So.
Okay. It may be gravid. It may have eggs
Bridge
underneath.
nav.
I
Its
think it
abdomen.
does look like there's something there. Looks like they're pretty well developed, our eggs there.
Hey, Nev. How's it going?
Do we have any more video?
Beautifully.
This association
we
with
have another
shrimp and
50 meters,
sponges
Another 50
is
meters.
found in the
Roger,
deep
roger.
sea and
Thank
also
you.
in shallow waters, so it's really common symbiosis between shrimps and sponges. Many different species of shrimps inhabit different species of sponge.
Don't know if we've seen this shrimp morph before, actually. Looking at it, it's hard to see the rock here from my view, but
So Tim is just noting that we may not have seen this shrimp morphotype before, and it's also interesting observation that this sponge morphotype is new to several of our eyes. So cool.
there's really antennae if you notice that.
Straight down and off track.
Could you pan back up to the shrimp?
Sorry. Thought I saw another associate down there.
Thanks.
Try to grab it. It's about time for us to leave.
Yeah, Andrew, can you have them just focus through, like, from the body to head? I kind of want to rostrum.
The rostrum.
Yeah, if it's possible.
Could you focus on the head anymore? Video request from Shoreside.
Couple more seconds, then I gotta run.
So we're at a depth of 2,741 meters, getting a really great close-up of this shrimp associate with this sponge. Both are new for this expedition and definitely haven't seen this before. Several sponges on top. Thank you,
Great.
pilot. Thank you, video. Again, possibly more of the tulip sponges, the I'm having a tough time saying Latin names today. I don't know why. Hyalonema. Okay, we're at a depth of 2,728 meters, continuing our transit upslope, surveying Exocet Seamount.
There's definitely some sort of trend in the rocks here. Again, we've moved up a little bit. D2 is facing almost directly due east, and so we can see that the trend of the rocks are essentially due east-west.
So this is the 11th dive of our expedition. Today is the 28th of April, 2015. Local time, 11:18. UTC, 15:18. Another shrimp in view, a couple more sponges, but not a lot of colonization of the rock faces today by sessile animals.
And lots of holothurian tracks.
Yeah, that's right. We were able to observe a holothurian making a track, right, the other day.
So Jeff, could we maybe zoom into those layers that are appearing in the bottom middle of the screen now? Just, I mean, again, we don't need to dwell, but just...
Of course. Just gonna get a little more light there for you. We won't dwell too long.
Thanks.
But, I have some time too, so not too big a hurry at this point. But yeah, we can come in. Just track along these layers.
Again, we can see the black color, which is the iron manganese. I know I'm saying that a lot, but it is what makes it so difficult. What interested me looking in there is we could see these layers. You can see the lines, the little outcrops of rock. I mean, they're only 10 centimeters high, are forming, striking from the top right to the bottom left. And again, these would be characteristic of some sort of bedded deposit, which could be sedimentary, or it could even be volcanic. If it's a pyroclastic flow, an ashfall deposit from an erupting volcano, we might expect bedding or layers too. But we're still trying to see some definitive piece of evidence. Thanks, Jeff. That's great.
When you guys come back out, there may be a fish.
Okay.
There's a possible fish sighting. So let's see if we... Randy, do you see it now? Okay, we can continue. Thanks. So again, I just want to mention, because we had such a great question, we had a Reddit AMA a week and a half ago, and had a really good question about organisms that may be more susceptible to anthropogenic disturbances in the deep sea because they live in harsh environment. And although we think that the deep sea is a harsh environment, we have to remember that these animals have evolved certain adaptations to survive this high pressure, low temperature conditions, and this includes some adaptations at the cellular or molecular level and also morphologically. So high pressure and cold temperatures can denature proteins, can decrease enzymatic activity, can decrease the fluidity of membranes. So go ahead.
Sorry, Jeff, can we zoom in on the stuff that's just going past on the right? Again, you can see this fabric, this layering, if that's at all possible.
It is possible, yep. Just a lot of inertia to stop.
Sorry, Andrea.
No, that's okay. Just chatting away.
All right, video, come on in.
I mean, again, I think we can clearly see convincing bedding here, or at least a foliation, a layering. Again, we see the lasers 10 centimeters, 20 meters, 20 centimeters thick. Just hoping that somewhere we can see part of the rock that's been broken, that we can see under the iron manganese coating. But definitely we are clearly seeing this 10 to 20 centimeter scale bedding. The other thing to point out, which is very obvious, is it's not horizontal, it's dipping into the hillside. So again, it's maybe hinting towards older rocks. The carbonate platform that we know of is relatively gently dipping in most places.
So we had a good shot of a purple holothurian, possibly a Benthodeides species, and several different sponges here as well.
That's great, Jeff. We can zoom out and just again get the bigger picture.
Hyalonema,
Yeah, video.
tulip sponges.
Thanks, guys. That was great. And again, this east-west trend, essentially.
We're at a depth of 2,715 meters. So just again thinking about what high pressure and low temperature can do at the cellular level, some organisms have evolved certain ways to combat the high pressure cold temps, including having elevated protein chaperones, so certain molecules that can help stabilize proteins and help with protein folding. They can increase enzyme concentration in cells. Some of the cell membranes have more unsaturated fatty acids, so this helps with keeping the membranes more fluid. And also in a lot of fishes, there's this osmolate called trimethylamine oxide, and this also is thought to help in stabilizing proteins. And interestingly, some studies have shown that this TMAO, it's called, increases in fishes that are found in deeper waters. So pressure and temperature really do control bathymetric distributions in deep sea fauna. And Todd, what is, co-pilot, watch lead.
Go ahead, watch lead.
Have you calculated our PSI today at this depth?
We can tell you it is 3,991 PSI at D2.
Thank you for that. And we are just less than half where we were in the Puerto Rico Trench a couple days ago, and the PSI there was about 8,900. Is that correct?
Yeah, 88 and change.
88 and change. Yeah, so I talked about some molecular and cellular adaptations, but there are also many morphological adaptations for combating high pressure, especially in fishes. This includes having more watery musculature, having no swim bladders or swim bladders that may be filled with lipids, and also reduced bone structure. So thanks for that last part, Randy.
We should certainly grab this.
Is this a swimming isopod, perhaps? Can we zoom in? So I think this is a type of swimming isopod. So, you know, a crustacean here, not a jelly or something like that. We often see these long tentacles coming off a jellyfish, so these are actually the legs of this isopod, if that is what it is.
Can't hear you. The end of his legs have...
Nav, this bridge?
Oh yeah, got little feather-like
Cilia. I just want to confirm, bailout position is still to the east?
Okay.
That's correct.
I'll call him silly.
And this would be a good time to extend another 50 meters, please.
Womp womp.
Roger that,
Thanks, pilot.
So, as we start to get ready for another watch change, it might be worth just stating the obvious in the geology, and other than what I've said a lot, which is it's hard, but we have seen a change. When we started off, we were seeing big blocks, rounded blocks, more massive outcrop, and for the last, I'm looking at my notes, since about 14:35 UTC time, we've been seeing this layering, these 10 to 20 centimeter east-west trending layers. So we have almost certainly found some change in the rock type from one type of rock to another. Question is, what are those rocks? And that's the real difficult question.
I'm going to follow the pilots and take a lunch break. I'll be back in 15.
Okay, guys, now we can potty. So more holothurian trails as we traverse upwards or along. Looks like sponges. Again on this slabby, layered outcrop. I'm guessing that's a little white sponge in the center of the field of view. So, Jeff, maybe we can try and zoom in again on these rocks just to try and get a feel for what they might be.
Absolutely. Let me just settle in for a really good close lit up view here for you. You can sort of take in moving blue water-ish views. Maybe this can help.
Okay. Hello, is somebody new on the line?
Hey Mike, Amanda Demopoulos from USG is here.
Hi, Amanda. How are you?
I'm doing well. How are you?
Not so bad, thank you. Now you're here, in a minute or two, I'm going to ask you about Seamount rocks.
All right, video. Come on in.
Just because I work for the USG.
No, because you work with Jason.
Yep, sounds good.
So we're just zooming in on here to again try and look for the texture of the rocks and the detailed biology for that matter. I think we can see these tiny little rods of shells. Are they pteropods or are they spicules? And a little sponge, I think, in the center of the field of view. We're doing a pilot change at the moment, so thanks, Todd.
All right, I'm going to lift off and hand over. Anything, Mike? Does this help at all?
I think that's good.
All right.
Jeff.
Video when you're ready.
Sorry, you guys are in control. Take over if you're about to change watches.
That's okay. I'll get the shot and then we'll change.
So Amanda, given that we are in fact changing pilots at the moment, can you tell us a little bit about the seamounts that you dove on with the Nautilus expedition further to the northeast of us?
Sure, it's been incredible to have the opportunity to dive here at Exocet. We've, as you mentioned, Mike, we conducted dives using the Hercules ROV both in 2013 and 2014 on Seamount northeast of us, where we are right now.
We are moving along at 0.1 knots.
That we explored included
at my heading
Norwa,
of 255.
Dog, and Conrad Seamount.
It's a steady move, and my
essentially,
camera
there hadn't
started
been
to aim
any
down.
ROV observations of those seamounts prior to our expedition, so we really were seeing the seamounts for the first time. And so there was a broad suite of different questions that we were interested in addressing. The specific questions included identifying the origin of the seamounts, whether they're from old volcanic origin or other sources, and then in addition we were curious about the ecology and the biology of these seamounts in this region, identifying the animals that were utilizing the seamounts as a habitat, and because we were able to cover quite a broad depth range, it gave us a perspective as to the community structure of the fauna residing on the seamounts at different depth intervals.
Sorry, Amanda, we'll come back.
That really expands our understanding of seamount habitat in the region. This dive is conducted. It started around 2,000 meters. It's far deeper than what we were able to conduct at the other seamounts in the northeast. So it's incredible to be able to take a look at this seamount at this depth range.
Thank you, Amanda. Sorry to interrupt. I thought there was something really interesting there. Just another holothurian, I think. But some nice close-ups.
See what's... that's a sponge.
And a sponge.
We've been seeing a few of these, what we call mobile benthic organisms, these holothurians or sea cucumbers on this dive so far. These animals are surface deposit feeders. They feed on the material that's, you can see in front of you, that sort of fine-grained material, and are important in terms of, really processing that carbon or energy in the deep sea. Also characteristic of this sort of sediment drape, the thin material that's coating the rocks, is the pteropod tests that you mentioned, Mike. Those are pretty common in this area. These are, planktonic organisms that once they die, their tests are calcareous, basically calcareous organisms, that material then settles out to the seafloor and makes sediment on the seafloor.
Thank you, Amanda. That was great. And the other thing I should just point out, we were just seeing it in the outcrop there, that there appeared to be another kind of weakness, another set of lines in the rocks, which might have been a cleavage. So we might have been seeing two things there. The bedding, which is approximately trending east-west, and then a steeper cleavage cutting that bedding. Or I should say more correctly, cutting that layering. If it is a cleavage, then it is indicating that these might be older rocks, metamorphic rocks. Typically a cleavage develops when rocks have been deformed. And so if we see another better example of that, I'll point that out.
Yeah, I'm still looking straight down at you. I think you're going a little slower than point 1.
Yeah.
Mike, is this stock crinoid on the lower left? I'm a bit delayed, so I guess that we just passed it.
Sorry, Amanda. I think we also need to catch up a little bit. Pilot Nav, yeah, you guys are in control in terms of getting to as far as we can today. Don't let us dawdle too much.
Roger that. Yeah, I've fallen back a little bit, so I have to push ahead.
Thank you.
And then we can take some nice close-ups. What's your camera angle? 40? Okay.
40.
So now we've come to smaller boulders. You can see the lasers are still on, so you can judge how big these boulders are, something like maybe 50 centimeters or 20 to 50 centimeters. And again, just trying to look for the elusive rock that's cracked open, that we can see what's inside under the manganese coating. So Josh, that's the instruction. Your eyes are much better than I. If you see a rock that looks broken, that's where we go. Is that a sea star we're just passing over, or another holothurian?
Possibly a sea star. I'm not far enough ahead, so I'll come down and
yeah. Let's take a look at that.
Peek.
Thank you, Josh.
Is it a star-shaped holothurian?
A star-shaped holothurian, yeah.
Hold on, video.
All right. Holothurian colored sea star. Amanda says it's a slime star.
That was a question mark. It resembles some of the slime stars that we were
come on in, video.
observing on some of our dives earlier in this expedition.
Full zoom?
Yeah, come out partial. Frame it, please.
Thing that maybe Chris Ma from the Smithsonian can help us identify.
Sure. So Chris, if you're listening.
You can come in tighter if you'd like video.
If only it would do something.
Can we zoom in any more, right to the top of the star?
Sure. If we have any more.
If we get the detail of...
That's max zoom right there.
So Amanda, that's its anus, is that correct?
Yeah, that's the backside, and its mouth is on the underside with the sediment.
Right.
Facing the sediments, essentially.
Drifting off.
It's quite a lot of detail on the sea stars. I've been really struck by all the different asteroid sea stars I've been seeing on the expedition so far. There are different morphologies, different shapes and sizes, and really patterns, something that I haven't really appreciated in the past. My work is mostly focused in the sediments, or what we can't see in front of us, but down